This experiment is about pollen germination for any flower, how you can view it in the slides, how the pollen tubes are getting germinated under the microscope. So again, before proceeding with the details related to the experiments, we'll define the strategy to always remember it, the steps, the instruments required, and what you need to observe. So the strategy is that just close your eyes, speak to your mind that you're going to learn about the experiment related to pollen germination, and you are going to create the visual image about the entire experiment by just listening to this audio, which will help you in recalling everything during the examination or whenever you want it. So let's begin with it. So we'll first define all the item list which we require to carry out the experiment. So we'll need a beaker, we need about boric acid, which is about 10 mg, magnesium sulfate half a gram, potassium nitrate half gram. You can use flour, any kind of flour. I've just mentioned here the name as hibiscus and vanca, anything which is available for your experiment performing. You need distilled water, you need sugar 10 grams, you need needle, a glass rod to make the solution, slide and the cover slip, you need the microscope and a dropper. So your equipment will be looking like, so you can have a look at the picture visualization in the slide so that you can create the image. So the steps for this experiment are quite simple. We have created a simple flow diagram to show you how you are going to perform the experiment. So we will need the stigma of the flower. So you can use any flower which will be provided during your experiment examination. You will need a solution to be prepared. We have defined the steps for the solutions subsequent slides. So what we would like to put down here is that you can use two types of solution, and the difference between these two solutions is that one will have just the sugar and the water dissolved completely in the water. The second one will have, along with the water, sugar, some chemical nutrients like potassium nitrate, magnesium sulfate and boric acid. So the difference between these two solutions is that if you are using only this sugar and water solution, then you will be seeing your pollen tube germination result in 30 to 35 minutes. But if you are using the other type of solution where you are using certain chemical nutrients, then you can see the result in a faster time, maybe between 10 to 15 minutes. That's the only difference. Otherwise, in both the solution, you will be seeing the pollen tube germination. So you will put the stigma on the slide using the needle and forceps. You will add 2 to 3 drops of any of the solution. Okay, so you will not use both the solutions. You can use any of the solution. We have defined the steps further. If you're using only the plain sugar solution, or if you're using the chemical solution along with mixed with the water and the sugar. Then you wait for that defined duration time, place a slide which is covered with a cover slip so that no other impurities and dust get settled over it, and watch it under the microscope. You will be able to see the pollen tubes. So steps. In this slide, we are going to provide you the steps where you will be preparing the solution using the chemical nutrients as well. So the first step will be you will prepare the solution. So take the beaker, add 50 ml of water. In that, add 10 gram of sugar. You can add 0.5 gram of magnesium sulfate, 0.5 gram of potassium nitrate, 10 milligram of boric acid. Now use the glass rod and mix the whole content in the water properly that it dissolves and you can get the transparent consistency. So it is crystal clear to see through it. Now move on to preparing the slide. Clean the slide. Now you transfer the pollen grains using the needle or the teaser from the flower. Once you can do that clearly, now add 2-3 drops of or 20% of this solution over that on the slide and cover the slide with a cover slip. Leave the slide for 10 minutes. After that, pick up the slide and put it under the microscope and view it. You will be able to see the pollen tubes which will keep on getting generated. So you can view so many of them based on the number of pollens you have taken, because a one pollen grain generate one pollen tubes. So here we are going to define the steps when you are just preparing a plain sugar water and going to generate the pollen tubes and see the pollen tubes. So there is no much difference. The only thing what you will be doing here is that you will not use any chemical nutrients. You'll just use your 50 ml of water and you will add your 10 gram of sugar and mix it properly that it get dissolved in the water. And again prepare the slide in the similar way, that you clean the slide, take out the pollens using a needle and tweezer as we have defined earlier. For the hibiscus flower, you can just directly see on the tip of it. For the vanca flower, you have to take out the petals. You'll see that green tender stick. Break that and take out those anther, and from the anther you can take out the stigma and place it onto the slide. Once you have done that, put some drops of solution over that and cover the slide so that there are no impurities. Leave this slide for 30 to 35 minutes just like that and then watch under the microscope. You will be able to see the pollen tubes. So this is how you will be seeing under the microscope the white transparent stick-like thing that is actually nothing but your pollen tubes. Once you visualize this under the microscope, then you can show it to your examiner. So these are some of the important tips which we have informed you before also, and we have put down in this slide. I'm just again repeating it, that if you're using only the plain sugar solution or sucrose solution, then you might have to wait for around 30 to 35 minutes to see the actual results under the microscope. And how to take out the stigma, so that's shown in the picture, that for the hibiscus you can see at the top of it, and we have defined that for the vanca flower you take out the petals, you'll see that tender green stick, break that, cut it, and then you can take out the anther, and from the anther using the forceps and the needle you can just take out the stigma. I hope this is clear and it will help you in the examination. Thank you.